Our project is about the diagnosis of lung cancer using 3D convolutional neural networks on CT images. Our group members are Xiang, Longfei and Xiang. The task of our project is to identify patients with cancer using 3D convolutional neural networks on 3D CT images. The dataset is 150 GB in DICOM format. It contains images of 1,600 patients. To prepare data for our 3D CNN model, we take these data pre-processing procedures. I'll talk about them in details later. Each patient has a folder named with the patient ID, which contains many DICOM files. Each DICOM file contains information for a single CT image slice. It has some useful information about the CT image. So we pre-process all the DICOM files in each folder and save these slices into a Python list. The list is ordered by the values of image position. Hounsfield unit is a measure of radial density. It can help us to identify different substances in the body. However, the original pixel values are not in HU, but we can calculate HU using this equation. The HU is equals to the pixel values multiplied by the slope values plus intercept values. These DICOM files come from different setting machines. They use different pixel resolutions, which cause pixel spacing inconsistency. Here I set the pixel resolutions to be 2 mm and make resampling. 2 mm can retain most useful information of the image. It leads to manageable file sizes. It can also help reduce dimensionality and noise. Next, I create a lung mask by setting a HU threshold. It can help to isolate lung tissues. Here I give an example of two 3D images to show that the lung tissues are selected out after masking. Some nodules are close to the edge of the lung, and they are hard to be captured, so I dilate the mask to ensure that all the nodules can be captured. Here I give an example to show that the wet area of the mask becomes larger after dilation, so that it can capture the nodules which are close to the edge of the lung. In the last, I crop the top and bottom slices as they are mostly blank, then resize the images to be 120 pixels in each dimension. Finally, I got 19.3 GB data.
This slide shows all the baseline models we implemented to compare against the XGBoost and the convolutional neural network. We can see that random forests and SVM are not picking up the underlying signal very well because their training and testing accuracies are fairly poor. They're also not predicting any positives at all. On the other hand, logistic regression is slightly better. It did well on the training set, but its false negative and false positive are still too high for the test data. XGBoost is a boosting algorithm. Its training can be accelerated using multiple threads in the CPU and also distributed across a cluster of Spark nodes. We gave it a try and see how it compares to CNN. We flattened the 3D images to 1D as an input, and then split the data into two sets to train up a bag of two XGBoost model due to memory constraint. The result is not very good, probably due to the loss of spatial signal during flattening. It is not predicting any positive cases for the testing data, just like the random forest and SVM. This is the CNN we built using TensorFlow. The top row shows the convolutional layers, the bottom row shows the fully connected layers. The idea behind is to slowly extract the 3D features from the sample in the convolutional layers, and then feed the features into fully connected layers for learning. This is the log-loss plot over the training epoch. It shows that our CNN model has converged at around 250 epoch. The CNN model was trained using the NC6VM with a Tesla K80 GPU, but it still took an extensive amount of time to train, so optimizing all the hyperparameters is quite impossible within the project timeframe. However, we did find that adding L2 regularization boosted the accuracy by about 3%. The final result is not stellar, but it is better than all the baseline models and the XGBoost. We can see that among them, the CNN is predicting the highest true positive and the lowest false negative for both training and testing data. This lung cancer classification is a hard problem. Some of the classifiers we saw previously had trouble predicting positive, as we only have 26% positive cases in the dataset. Also, the dimensionality is huge while the training sample is small, rendering the models to easily overfit. And the inputs are noisy even after masking. The noises can include the small intestine and lower abdomen, air bubbles, blood vessels, and anything that are basically not cancerous nodules. So to further the work, we could download some pre-trained CNN and train upon them, or manually extract relevant features using domain knowledge and pixel manipulations on the images. And of course, obtain more data.